So we all see the growth characteristics. Yes?
Yes. Yeah.
So we are going to look at some growth characteristics for bacteria growing on agar, as well as for bacteria growing in broth. And so the agar characteristics you need to pay attention to, because you'll be using some of those next week on the unknown. And I'll talk a little bit about the unknown today, but more next week. Just nothing to worry about. For the broth characteristics, you need to know those, especially for the lab exam. So we are going to determine if bacteria produce a definite pigment and where that pigment is located. We'll also look at the consistency of bacteria, and we focus on just two consistencies, mucoid or dry. And then we'll look at broth characteristics like turbidity, pellicle, flocculent, and sediment. So for the agar slant, pigment is something that's very interesting. Not all bacteria produce a definite pigment. It's just they're not invisible. If you get millions of cells together, you can see it. But if you ever want to describe the growth as kind of whitish, off-white, or buff-colored, then it's most likely not a definite pigment. If the growth is bright white, then that would be considered a pigment. But some bacteria do produce very colorful pigments, and the question is, why do they do that, right? It's not just to look pretty, because pigments are metabolically expensive to produce, because the bacteria have to use ATP to synthesize these pigments, so they must serve a purpose. And so some pigments are photosynthetic, although most of them are not, but some pigments are used for photosynthesis. Some pigments are involved in the pathogenesis of these organisms to help make them pathogenic. Some of these pigments help to absorb UV light, which can protect them from damage caused by UV. Some of these pigments are toxic to predators, so there are protozoa like paramecia that love to eat bacteria. And so eating some of these bacteria will kill that paramecium. So these pigments can be toxic to predators. They can serve as antioxidants, right, to neutralize free radicals. And some of these pigments are actually antibiotics that certain bacteria use to inhibit or kill other competing bacteria. So the pigments do serve a purpose, not just to make the bacteria colorful and pretty. Now, these pigments also have some potential human uses. So we know that some of these pigments kill other bacteria, so there's the potential of using these pigments as antibiotics. We can use them as antioxidants, right, to neutralize free radicals inside of us. Some of these pigments have shown some anti-cancer properties. And since the pigments are colorful, we can use them as coloring agents, right, to color textiles and cosmetics. So there are potential uses for these pigments, and they serve a purpose in bacteria. So as I said, not all bacteria produce a definite pigment, but those that do, the pigment is either inside the bacteria, which makes it an intracellular pigment, or the pigment is secreted into the external environment, making it an extracellular pigment. So it's usually where the pigment is. It's inside the cells or it's outside. But there are always exceptions. There are certain bacteria like Serratia marcescens, strain D1, which both has lots of pigment inside the cells, but it also secretes the pigment to the external environment. But Serratia marcescens D1 produces a deep red pigment. It's actually very pretty. But that pigment is both inside and it's also secreted to the outside environment. So here we see some examples of some bacteria growing on the agar slants. And so let's start from the right and go to the left. So Micrococcus luteus. What do you see here? Micrococcus luteus. Is there a definite pigment?
Yes.
Yes. And what's its color?
Yellow.
And where is it? Intracellular or extracellular?
Intracellular.
Intracellular, right? So we see the yellow growth on top of the agar. Now the agar itself kind of has a slight yellow color to it, but that's just the color of the agar. You always want to look at the growth on the agar. So Micrococcus produces a definite yellow pigment that is intracellular. What about Cocuria rosea? Is there a pigment? You got to speak. I can't see any chats because I'm looking at the PDF. So is there a definite pigment?
Yes.
What color?
Pink.
Pink. Intracellular or extracellular? Intra or extra?
Intracellular.
Intracellular, right? And next we see that beautiful serratia marcescens and that deep red pigment. Is it intracellular or extracellular?
Did we just say it was both?
Right, right. But based on what you can see, it looks intracellular, right? Because we're looking at the top down. If we turn the agar slant sideways so we could see into the agar, we would also notice red moving into the agar, but we can't see it from this angle. So it looks like it's intracellular even though it's both. Chromobacterium violaceum. Well, that name tells you the color, right? Violet. And it's such a dark purple, it almost looks black. So a definite pigment here, intracellular or extracellular?
Intracellular.
Intracellular. Next, we have Pseudomonas aeruginosa. And it is hard to see on the slant. Ideally, we need to turn the slant sideways so we can see the agar. But if you kind of compare the agar in the tubes next to it, you see that this agar is greenish. It's got a greenish tinge to it. But the growth is just kind of grayish looking. So there's no definite pigment in the bacteria. But the pigment is being excreted into the agar, which is giving it that greenish color. So Pseudomonas does produce a green extracellular pigment, but the growth itself is not pigmented. And then here we have Staph epidermidis. Is there a definite pigment?
Yes. White.
Yeah, right. Because this is white, right? The Pseudomonas is not white, but this is definitely white. So if it's a bright white, then it's a pigment. If it's just kind of whitish, right, that's not a definite pigment. So any questions on the pigments? So for consistency, there are technically 3 consistencies, but we only use 2, mucoid and dry. When you hear the word mucoid, what do you think of? What do you think of when you hear mucoid?
Mucus.
Mucus, right? And mucus is slimy, shiny, wet looking. And so most bacteria are mucoid. Some are extremely mucoid, others not so much. It's the not so much that has the other category called butyric, but we're not going to use that. But if you sort of think of like a creamy, buttery consistency, that would be butyric. But we're calling that mucoid, just not very mucoid. A few things are dry. So dry is not shiny, not slimy, not wet. The fancy word is friable because the bacterial growth looks very crusty, crumbly, like you could easily break it up into pieces. Not many bacteria are dry. Most are mucoid, right, to some degree or another. Now, when it comes to our bacteria here, it's always important that you look at the growth, not the agar surface, because the agar surface is naturally shiny. But you want to look at the growth on the surface. And so you can see how the growth on Micrococcus is kind of shiny, right? So that would be mucoid. On Cocuria rosea, again, it's kind of shiny, shiny, shiny. It's hard to see on the Pseudomonas, but it is shiny. It's just not very shiny, but it is. And it's something with Staph epidermidis, it's shiny. So all these are mucoid. We don't have any pictures of dry growth on the agar, but if we looked up Mycobacterium on agar, we would see that it's dry. I don't know if I can find. Yeah, here it's, you can kind of see here, right? It's kind of dry looking. So it's not shiny or slimy looking. It's kind of dry. And you also notice something else. What else do you notice about Mycobacterium? What else do you notice here? We don't see anything else there?
It's orange.
Right, so there's a definite pigment. It's kind of a golden, yellow, or orangey pigment. But that was an example of a dry organism. So just summarize, and I'll try to write here. So we did not see Bacillus cereus, but Bacillus cereus would look kind of whitish. So is that a definite pigment?
No.
No. So Bacillus cereus, no, right? So there's really no color because there's no definite pigment, no location because no definite pigment, and it does look kind of shiny, so that would make it mucoid, right? Now, Chromobacterium violaceum, did that have a definite pigment? The Chromobacterium violaceum?
Yeah, it did.
Oh yeah, right. And what color was it?
Was it purple or black?
Very dark purple, right? Was it intracellular or extracellular?
Intracellular.
Which cellular?
Intra?
Right, intracellular. And the consistency?
Mucoid.
Mucoid, right? Now, Klebsiella pneumoniae, again, no picture shown. It, again, is kind of whitish looking. So typically, we say there's no definite pigment, so I said no color, no location. And a Klebsiella is very slimy. So it's very, very mucoid. Micrococcus luteus. Was there pigment?
Yes, it was yellow.
Yep, it was yellow. That's a Y. Where was the pigment?
Intracellular.
Intracellular. And mucoid or dry?
Mucoid.
Mucoid. Mycobacterium flea, I just showed you a picture on the internet. Is there a pigment?
Yes.
Oh yes, right. It was kind of golden yellow. It's supposed to be golden. Right, the pen doesn't pick up, so a golden yellow. Intracellular or extracellular?
Intracellular.
Intra. And mucoid or dry?
Dry.
Dry, right? Or the fancy word friable. And the Pseudomonas aeruginosa, which was in the picture, was there pigment?
No.
Oh, yes, there was, right?
Oh, there was?
There was. I don't know why this thing does this.
Oh, it was green, but it was
Yeah,
outside. Oh,
right.
sorry.
So it's supposed to be a G. Yeah, see, the pen is weird. And it won't let me write, the right thing. So, but it's green. And it was, we said extracellular. Let's see. And dry or mucoid.
Mucoid.
It's mucoid. It's just not very mucoid. Right. So those were the characteristics for the agar. When it comes to broth, we have 4 characteristics we look at. So if the bacteria are growing in the broth, it makes the broth cloudy. And the fancy word for cloudy is turbidity. So any degree of cloudiness means the broth is turbid. So if it's only very slightly cloudy, it's still turbid. There's still growth in the broth. When bacteria die, they fall to the bottom and accumulate. So we call that sediment. Remember, we looked at turbidity and sediment when we did the temperature experiment, and we're looking for growth in the tubes. Some bacteria do actually grow as a sediment, but mostly it's just dead cells that fall to the bottom. A pellicle is a layer of growth across the surface of the broth. So the bacteria can walk on water, and so they grow across the surface. Now, you may remember water in a tube. It kind of bends at the top of the tube, and we call that the meniscus. And especially with broth, which has kind of a yellowish color to it, it looks darker at the meniscus. So that's no growth. It's just the way the liquid looks in the tube. So it's important not to confuse a pellicle with a meniscus. And then some bacteria will grow as clumps or clusters that either stick to the sides of the tube or that float around through the broth. But those are the 4 characteristics. So here we can see a pellicle, and this happens to be Mycobacterium flea, which forms a very thick pellicle, and then the bacteria start to grow up the sides of the tube. There's no turbidity, right? The broth is clear, but there is some sediment, and that comes from the pellicle breaking apart and falling down to the bottom. Here we can see the broth is cloudy, right? So there's turbidity and there's sediment. And here we see a meniscus, right? So this is where the water is bending. It looks a different color because of the light that's being refracted, but it's just a meniscus. There's no growth, right? The broth is turbid and there's sediment. And here we see other little clumps of bacteria, so this would be flocculent. Now, a broth culture can have more than one characteristic. And so I'm going to try and draw something. So I don't know what that thing is there, but so here we have a tube. So what three characteristics do we see there?
Sediment
Sediment,
and a pellicle.
pellicle, and this is my attempt at turbidity. Now, if this were the Pseudomonas tube, it's not what I wanted to do. Go back. So let me see now. Can I change that? Nope. Like I said, it's very hard to draw in a PDF, and my tablet doesn't want to respond. So let me see now. Can I do it? Nope. Yeah, like I said, it's very tedious in a PDF. I don't want. What I'm trying to do is be able to draw green. Let me, all right, maybe. Oh. Yeah. All right, it's not going to work, so I can't do what I want to do in this. But what I'm trying to do is to draw some green without making the whole thing green. Oh, okay, now I can do it. So if this were the Pseudomonas tube, this is what we would see. We have pellicle, sediment, and turbidity, and then we see something else there. So what is this? So what is that?
A meniscus?
Nope, no meniscus because we do have a pellicle. So what did we say was special about Pseudomonas aeruginosa on the agar slant?
Is that the extracellular pigment?
Right, so you would actually see the broth turning green just under the pellicle because there's a lot of bacteria there and they're secreting the pigment. So you can also see pigment in a broth culture. But the four primary characteristics we look for are turbidity, sediment, pellicle, and flocculent. But it's possible for a broth tube to have more than one characteristic. So you just need to be able to recognize the characteristic when you see it. You don't need to know which bacteria have which characteristic. So we had no Bacillus cereus, but it's cloudy. So that's supposed to be a check. I told you this thing. So I just need to give up with this. All right, so it's cloudy. It's not letting me draw. Draw. Okay. So that's supposed to be a yes. There's stuff on the bottom. I'm ready to give up on this because it's just not working. Nope, it does not want to let me do anything. So yes, there's sediment, no pellicle, no flocculent, no pigment. No, no, no. Yes, but it won't let me make a Y. Chromobacterium, yes to turbidity. Yes. 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 Klebsiella, yes. Turbidity, yes. Sediment, no pellicle, no flocculent, no pigment. Micrococcus luteus, yes. Turbidity, yes. Sediment, no pellicle, no flocculent, yes. Pigment, right? That yellow pigment. Mycobacterium flea, no turbidity. There may be sediment if the pellicle breaks. Yes, a major pellicle. No flocculent. Yes to pigment. And the Pseudomonas, that's the one that I drew for you. So yes turbidity, yes sediment, yes pellicle, no flocculent, and yes pigment. So you just need to be able to recognize the characteristic in broth when you see it. On the agar characteristics, you should pay attention to what the actual bacteria look like, because it's possible you could get one of those for your unknown. But let's finish reviewing this, and I'll talk a little bit about the unknown.
What's a flocculent again?
Little clusters of bacteria sticking to the sides or floating to the broth.
Okay, thanks.
So do all bacteria produce a definite pigment?
No.
No. But if they do produce a definite pigment, where can you find it?
Inside or outside the cell?
Right. So if the pigment is still inside the cells, it's an intracellular pigment. If the pigment has been secreted, it's extracellular, but it's all relative to the cell. And so why do some bacteria produce these very colorful pigments? Is it just to make them look pretty?
It could be for photosynthesis.
It could be for what, Craig?
Photosynthesis.
Right, for photosynthesis. It could be for protection, right, from UV, from predators, uh, antioxidants. So there are reasons why the bacteria actually make the pigments. And so what can humans do with those pigments? So how can humans use these pigments? Some of these pigments may be natural antibiotics we could use. We could use them as coloring agents for things, right? So there are potential uses for these different kinds of pigments. And what were the two growth consistencies? And describe them?
Was it the dry and the mucoid?
Yep, dry and mucoid. And what does a mucoid look like?
Mucoid is shiny and slimy.
Right, shiny and slimy. And the dry?
Is not slimy.
Right.
Friable, is that the word?
Yeah, they look rough and crumbly, like they would break apart. And to go back to mucoid, I just want to show you something which happens in an agar slant. And so, because agar is actually mostly water, we often get condensation. So you can see there's some liquid that has accumulated in the bottoms of these slants. So it's important to ignore that, right? That's just, water that's accumulated. Whenever you're looking at the agar characteristics, you look at the growth on the surface of the agar, so not the liquid that has accumulated in the bottom of the slant. And for the broth characteristics, we have turbidity, sediment, pellicle, and flocculence. Right? See the little clumps of bacteria here? So, you want to know something else that we can do with these pigmented bacteria? Nerds can be very artistic. Are we seeing my browser screen here?
Yeah, we can see.
So look what you can do with pigmented bacteria. You can make art. So these are images of pigmented bacteria growing on agar plates. And they actually have competitions where people draw the bacteria and let them grow, and then they produce all these amazing looking works of art using bacteria. I mean, look at these things. Have you ever seen these before?
No, it's very cool.
I mean, and like I said, they have contests. And people win, sort of awards for different, plates that they create. I mean, this one won an award of the American Society for Microbiology, their 2018 competition. This won first place. I mean, look at that. And the amount of time and effort needed to carefully add the bacteria to the plate and let them grow up. This is pretty amazing, right? So agar plate with all these bacteria growing on it. See the ocean waves. And of course, they can use genetically modified bacteria like with the P. glow plasmid and make them fluorescent like this. So what animal do you see? Do you see an animal? Here? It's a bunny rabbit, right? And look, here's a map of the world, all the countries. I mean, it really is pretty awesome, some of the things that have been done, all these different bacteria. Unfortunately, for some of these, we probably cannot use them in lab because technically they're a little more pathogenic than we're allowed to play with. But it would be cool if we could do that. Here in the United States at night, right? All the lights from space using fluorescent bacteria. So, I mean, it really is amazing what they have done with all these different things. Impressionism. Yeah, somebody's mouth. So, of course, you can't actually hang these on your walls, but you can take pictures of them. But look at what can be done with certain bacteria. It really is amazing. So, nerds make art, right? So, pretty awesome, actually. And then you can blow up the images and mount them on the wall. Some abstract art. So it's pretty cool, what they can do using bacteria, right, growing on agar. So anyway, so that was a fun thing. So for the unknown, I'll talk more about it on Monday, but basically you'll just be looking at pictures of things and then describing them, because normally what happens with the unknown is you're given a slant culture. You make observations of the growth, like is there pigment? If so, where is it? What color is it? Or there's no pigment at all? Whether it's dry or mucoid. And then you would make a gram stain, a spore stain, and an acid-fast stain, and interpret that. Is it gram positive or negative? Are there spores present? Is it acid-fast positive or negative? So I'll be giving you the images to look at to interpret. Is it a rhodcoccus or spiral? Is it in clusters, chain, single? And then you have to take all those data and make a logical guess as to what it could be. And so, you can use other resources to, look up and see what this organism could be. But you do have to give a genus and species that is logical for all the results. And normally this would all be done in 2 hours. So you'd actually make all your slides and do all your work and then turn it in before you left the lab. So I'll give you 24 hours to turn it in because you really don't need that much time. But that's how we're going to do the unknown. So on Monday, we'll talk more about it, and then I'll just email each of you your unknown sheets with your pictures on it to interpret. And so that's how we're going to do the first unknown.
So we can use our notes
Yep,
for this.
yep. That's what you were, in lab, you're allowed to use anything except each other. So you could use your books, your notes, the internet. That was fine, but you couldn't talk to each other. So
Got it.
it was a very quiet lab. All the
Okay.
students are all busy working. But that's how we're going to do the first unknown. So I'll just give you pictures of things like what you would have seen and done, and then you interpret them and then give me an identification that matches. So just, there can be different organisms, that match the same thing. It just has to be logical, for what's there. So no bizarre bacteria, no dangerous pathogens. It has to be the kind of thing that we would give you in lab.
Would it probably be something we've spoken about, like something familiar?
Yeah, right. In lab, it's typically something that you've seen already or that we've talked about. So again, no bizarre bacteria, right? No weird thing that no one's ever heard of, or some nasty, dangerous pathogen, right? It has to be something logical that we would give you. Although some of you, I might give a pathogen, right? Just kidding. So any other questions about anything? Okay, so remember, on Monday we do control of microbial growth, and then we'll talk about the unknown, and then I will email the unknown to you. No, the unknown's not on the lab exam, but everything that we've learned about is on the lab exam, and you apply that to the unknown. So all the material that you need to know and interpret, yes, that will be on the lab exam. But the unknown was intended to be a practical, test of your skills and abilities in the lab. Well, we can't do that. I'm just giving you pictures to interpret, but for the lab exam, yeah, you need to know what a gram stain is, how it works, all the stains involved, what gram positive, negative look like, things like that. So all the stuff that you're applying on the unknown, you'll be tested on the lab exam. And then on Wednesday, we'll just kind of have a review, right? Because we're ahead by one lecture, so we'll have a review for lecture exam 2 the following week. And then in lab, I think we're doing UV and maybe something else. So, but we're almost at midterm, right?
Wait, the lab exam is a week from today, right?
No, the lab exam is actually the week after, isn't it? It's not next week, the week after, I think, isn't it? Along with your lecture exam? Or maybe, or is it on Wednesday? Maybe it is on Wednesday. Oh, the 14th, so it's next week, next Wednesday. Okay, so
That's next Wednesday.
yeah, so we'll have the review in the lecture, and then we'll do the lab exam. And then the week after, you have your lecture exam too. So, all right, are we good?
Yes, thank you.
All right, have a good weekend. I'll see you guys on Monday.
You too. Bye-bye.
Bye-bye.
Professor, goodbye.
Bye, Clarice. Bye, Christina. Bye, Bryant. Bye, Christiana. Bye, Maria.